I call this meeting of the Senate Homeland Security and Government Affairs Subcommittee on Emerging Threats and Spending Oversight to order. I want to thank Senator Hassan for allowing this bipartisan hearing to occur. Welcome to each of our panelists. Thank you for joining us. The purpose of this hearing by the Subcommittee on Emerging Threats and Spending Oversight is to discuss, as our name implies, the emerging threat posed by gain-of-function research. We will hear from a panel of 3 witnesses, all of whom are extraordinarily accomplished experts in the scientific community. We're grateful for their work, and we are grateful to each of you for taking the time to appear with us this afternoon. Dr. Stephen Quay is the founder and chairman of the Seattle-based Atossa Therapeutics. Atossa Therapeutics is a clinical-stage biopharmaceutical company that develops novel therapeutics and delivery methods for breast cancer and other breast conditions, with the goal of preventing the 2 million yearly breast cancer cases worldwide. Earlier in his career, Dr. Quay received his MD and PhD from the University of Michigan, trained as a postdoctoral fellow at MIT, and served on the faculty of Stanford University School of Medicine. Dr. Quay published contributions to the world of medicine have been cited extensively, and he is a medical entrepreneur. He has founded 6 startups, invented 7 FDA-approved pharmaceuticals, and is the holder of 87 patents and over 130 pending U.S. and foreign patent applications. He's also an author. Notably, during the pandemic, Dr. Quay published his #1 Amazon bestseller, Stay Safe, A Physician's Guide to Survive Coronavirus. Finally, Dr. Quay recently presented testimony to lawmakers as a part of an expert forum convened by the House Select Committee on Coronavirus titled Led by Science, the COVID-19 Origin Story. Dr. Quay?
I'm honored to participate with my esteemed colleagues, Dr. Quay in this forum entitled Revisiting Gain-of-Function Research, What the Pandemic Taught Us, and Where Do We Go From Here? I offer 6 statements in opening. Very good. I offer 6 statements in opening. 1, there is no dispositive evidence the pandemic began as a spillover of a natural virus in a market. All evidence is consistent with a laboratory-acquired infection. I do understand this conclusion is not widely held, and I could spend an entire hearing painstakingly going through the scientific evidence for this conclusion, but that's not the purpose of today's meeting. I'm happy to discuss the evidence contained in my written remarks during questioning. I'm also willing to publicly debate any virologist on this question at any time or place. Only one infectious disease doctor was willing to debate this question with me last year in a formal debate format, and he lost. I'm also willing to testify under oath if requested. Number 2, all evidence is consistent with an accidental and not a deliberate release. Number 3, SARS-2 has features consistent with synthetic biology gain-of-function research. 2 features involve acceptable academic gain-of-function research: the receptor binding domain optimization and the furin cleavage site. These 2 features have never been found in nature in related viruses that could have reasonably started the pandemic because of the closeness of these viruses to Wuhan. These 2 features are, on the other hand, routinely engineered into viruses. In 2018, U.S. and WIV scientists proposed inserting, quote, human-specific furin cleavage sites in a bat virus backbone. 2 years later, SARS-2 appeared on the WIV doorstep. SARS-2 is a bat-derived virus with a human-specific furin cleavage site. One region of SARS-2, called ORF8, has features of forbidden gain-of-function research, asymptomatic transmission, and immune system evasion. The WIV was engineering a protein related to ORF8 to have these 2 forbidden properties before 2019, as shown in 2 master's degree theses available only in Chinese. COVID exhibits 40% asymptomatic transmission, unheard of for a new respiratory virus. Patients infected with an acquired deletion of ORF8 have a milder infection. Could the reduced efficacy of vaccines and natural immunity be an engineered feature? It appears likely. 6, in December 2019, the Wuhan Institute of Virology was conducting synthetic biology research on the Nipah virus, which is 60% lethal in low-containment BLSA 2-3 facilities. The Nipah virus was in an infectious clone format. Nipah is a BLSA 4-level pathogen and a CDC-designated bioterrorism agent. This is the most dangerous gain-of-function research I have ever encountered. We should assume this research continues to this day at the WIV. I'll close with 5 recommendations for future gain-of-function research. Where did the pandemic begin? The competing hypotheses are a natural spillover at the Hunan seafood market in Wuhan and a laboratory-acquired infection. 2 recent papers purport to claim the pandemic began at the Hunan market in December 2019. There are at least 6 serious problems with these papers. The most important are that in the early months, no animal has ever been found to be infected with COVID-19 anywhere, including the market, and the molecular clock of SARS-2 places the first human infection in the fall of 2019, long before the December market cases. And all infections in the market in humans were what's called lineage B and not the most ancestral lineage, lineage A. I, like many other scientists, believe the market cases were a super spreader event. This first chart here. The earliest cluster of hospitalized patients with both the lineage A and B virus was at the People's Liberation Army Hospital in Wuhan. This hospital is about 6 kilometers from the WIV and on line 2 of the Wuhan subway system, as shown in this chart. All early cases are in hospitals adjacent to line 2, and the probability this was a chance occurrence is 1 in 68,000. The line 2 COVID conduit, as I called it, includes the PLA hospital, the WIV, the market, and the international airport. You can literally walk down into the subway system from the WIV in China and next exit outside in London, Paris, Dubai, Los Angeles, or New York, all before having any symptoms. Modeling by others suggested the pandemic could not have occurred without the international spreading impact of line 2. Has gain-of-function research been useful to the COVID response or any other public health infectious disease emergency? I have found no evidence that gain-of-function research helped in either the COVID pandemic or other smaller epidemics. We now know that an MRI vaccine can be designed within literally days of a new outbreak once the pathogen is sequenced, and large-scale manufacturing can begin soon thereafter. This capability has now been fully road tested and provides, in my opinion, the best defensive capability against future microbes. It's also important to point out that gain-of-function research is a tiny sliver of all research funded by NIH. Specifically, there were over 36,000 R01 grants funded by NIH in 2020, the latest year with statistics. Of these, the self-described gain-of-function on potential pathogens research grants numbered only 21 in the latest funding year. Even expanding this by tenfold with a less stringent definition of gain-of-function would mean we are talking about less than 1% of all NIH research funding. I cannot imagine a scenario where, but for this tiny research effort, a new pandemic occurs. What reforms should be considered in order to assure that such research is conducted in a safe and transparent manner? While I found no actual benefit of gain-of-function research, I believe efforts to ban it, given the vested interests of literally the entire virology community, is a hill too steep to climb. A proposal that I believe is achievable is a placement of all select agent research within the existing institutional review board structure used for human clinical trials. I believe that this effort would put guardrails around the most dangerous aspects of this research and has the added benefit of international acceptance, including in China. My second reform would be to separate government oversight from the funding agency, and the model would be the Atomic Energy Commission. Third suggestion is to place Western biotechnology equipment under export controls and monitoring. There are ways to build into these systems a forensic and law enforcement capability that could, for example, with probable cause and a court-issued search warrant, allow the work of any lab in the world to be scrutinized remotely. My fourth recommendation is simple. Don't put dangerous infectious disease laboratories near subways like Line 2, where every major city in the world is accessible within the incubation period of an infection. Finally, including what I call going of opportunity research, going into caves where humans are seldom found, taking a bat fecal sample containing thousands of viruses, bringing those viruses back to a laboratory and culturing the specimens where a virus might be controlled in a diverse natural environment, is now able to grow unrestricted in pure culture, provides an immense increased potential risk, a gain of opportunity for the virus. This is the goal of the Global Virome Project, a Gates Foundation-funded EcoHealth Alliance-associated effort. Their stated goal, collect the estimated 500,000 unknown viruses that are capable of infecting humans and bring them back to a laboratory near you. What could go wrong? Could I have the last slide here? What happens if we have these hearings and nothing happens? In December 2019, we performed a remote audit, forensic examination of the Wuhan Institute of Virology and found synthetic biology experiments with the Nipah virus. As the chart shows, they had created a cloning vector with a virus the US CDC defines as bioterrorism agent. Nipah virus is one of the deadliest on the planet with a greater than 60% lethality. Why were they conducting this experiment? I do not know. But a laboratory-acquired infection with this virus, if it became airborne, would make COVID-19 look like a walk in the park. The work of this committee is critical to protecting the American people as well as the people of all countries from future pandemics, man-made or natural. If we now fail to act with the knowledge we have, history will judge us poorly. Thank you for the opportunity to speak. So,
was there a specific incident or something that concerned people that caused the pause?
Yes, there was experiments in influenza in the Netherlands and Wisconsin that took a virus that was 90% lethal but not airborne and made it airborne through passage in the laboratory.
And that occurred when?
2013, 2012.
And that caught the attention of who? I mean, who was alarmed by that and instituted the pause? I know it occurred under President Obama, but which member of our health agencies that?
Yeah, I think Dr. Ebright would be the best to answer that.
Dr. Ebright.
So the proximal impetus for the pause was a series of events, laboratory accidents at federal laboratories that have access to and storage of potential pandemic pathogens. The accidents included an anthrax incident at the CDC, another anthrax incident at a U.S. Army facility at Dugway in Utah, and the finding of unsecured vials labeled smallpox virus in an FDA NIH freezer in Maryland. And those three incidents occurring in close succession resulted in a hearing in the House Energy and Commerce Committee, and then action by the Office of Science, Technology, and Policy. So the pause was driven ultimately from the White House, from the Obama Office of Science, Technology, and Policy.
So listening to your testimony, I'm assuming all three of you would agree with this statement that this research, and I would say even the mining of dangerous potential pathogens, go crawl in a bat cave to try and pull these things out and bring them into a lab. There's certainly no benefit that overrides the risk. We shouldn't be doing this at all.
Yeah, I call it gain of function and gain of opportunity where you bring a virus back. And as I said, my analysis is that it hasn't contributed to the response to this pandemic.
So we shouldn't do it. I mean, we shouldn't be, we can talk about controls, but bottom line, we shouldn't have controls because we shouldn't even do it.
Thank you, Mr. Chairman. Thanks to the witnesses for being here. Dr. Quay, if I could just start with you. You said in your written testimony that the genome of COVID has some of the hallmarks of gain-of-function research, and in particular, 3 genomic regions you say have the signature of synthetic biology. 1 region has features of the 2 types of forbidden gain-of-function research that are associated with bioweapons development. And you said in your opening remarks that you believe COVID-19 was the product of gain-of-function research and was from a lab leak from the Wuhan Institute of Virology. My question, I guess, is, do you think that China engaged in a cover-up to prevent the world from knowing the true origins of this virus and a lab leak?
I think there's abundant evidence that they have not shared all the information they had at the time. They continue to not share information. I could give you a laundry list of 20 things that they've done, starting with a website with 21,000 viruses on September 12th. 2 a.m., someone was in the Wuhan Institute of Virology. That had been available to virologists for a decade. It was taken offline. It's not been returned. We've asked to see it. No one that I know of has ever seen it. It goes on from there.
Are you concerned with the continuation and expansion of Chinese gain-of-function research?
Well, I think I testified here that in December 2019, they were doing synthetic biology on a cloning vector of the Nipah virus, which is 60% lethal. We just experienced a 1% lethal virus. My estimates would be that could set us back a millennium. The Black Plague was a 20% lethal event, and it was 250 years for civilization to return.
Let me ask you this. How safe were the testing conditions at Wuhan, to your knowledge?
Well, I think that a lot of the Western virologists actually used the findings of that as a way to get around saying it was okay at the beginning. All of the work that I've described as being done at what's called BLSA 2-3 level, which is commonly spoken of as a dentist laboratory level of biosafety. So maybe a little higher than that, but that's not a bad euphemism.
You said, I think in your testimony, this is the most dangerous research that you have ever encountered. What makes this particular research so dangerous?
If you're doing experiments with a pathogen that is 60% lethal but is not airborne, and you make it airborne in the laboratory, and someone walks out with it. Nipah has a 21-day incubation period. It's perfect for widespread spread without being detected. We couldn't afford we can't afford 10% lethality.
Yeah.
So I guess my question, I think it's pretty clear, but I'd like to go through everybody, even though Dr. Ebright has said this was gain of function, to each of the three witnesses. Was the research where you take the backbone of a SARS-1 virus that has known lethality and you mix it together with an unknown bat virus S protein genes to create a new virus? Was this gain of function according to the NIH definition, and should it have been reviewed and discussed by this committee that was supposed to prevent dangerous research from going on? We'll start with Dr. Ebright. Dr. Quay.
So the Wuhan Institute of Virology is unique in the entire world. Before 2019, 65% of all publications on coronaviruses came from that single institution. They're unique for two reasons. For almost a decade, they were going into bat caves throughout China and actually back into Africa as well, 20 visits a year, and bringing these samples back to the laboratory. So on the one hand, they had the largest collection of raw material backbones from nature to then do gain-of-function research on. They trained in Galveston, Texas, and in North Carolina, and were doing experiments, published experiments between 2015 and 2019. And I believe it's the confluence of those two activities, gain of opportunity, bringing things back from bat caves, and gain-of-function research that led to the pandemic.
Now, people have said, well, the closest relative that we found is only 96% identical to COVID-19. This couldn't have come from the lab. They've also mistakenly accused those who say it came from the lab of saying, oh, it came from this particular variant. But I think what people who are saying that this could have come from a lab are saying that there could also be possibly other viruses that are closer that were manipulated, or that the one that is 96% analogous to COVID-19 could have gone through serial cell culture and become COVID-19. I guess I'd like to ask each three of you whether or not the variant that is 96% analogous to COVID-19, could it through serial passage be transformed to COVID-19? Is it possible? Is it so far away that you can't do it experimentally? Could you do it through gene splicing? Could it be done? Or is it something that argues that this couldn't have come from the lab? We'll start with Dr. Ebright.
The three sets of viruses that are closest to SARS-2 are one from southern China, RITG13, and a series of Banal from northern Laos. And as indicated, there are probably 1,200 letters different in the whole 30,000 letter alphabet. In nature, that takes approximately 40 years, so the most common ancestor is about 40 years ago, but most of that can be done in a couple days in a laboratory. However, I don't believe we currently have the starting material, the backbone on which SARS-2 was found. I think it's one of the other 21,000 viruses in the database that was taken down at 2 a.m. September 12th, 2019.
So a great deal of information was destroyed by the Chinese.
It was taken offline, not available. I don't know if it was destroyed.
Right. So what I'd like to ask, and I'm going to finish with this, and then we'll have another round if some people would like to ask some other questions, is Dr. Quay, could you sort of lay out in as simple a fashion as possible 2 or 3 items about the virus that makes you think it came from, and I don't think anybody knows with 100% whether this came from a lab or whether it came from animals, but there is some compelling evidence that suggests it could have come from the lab. And, you know, even if it was a 10% chance it came from a lab, it's another reason for us to be concerned about having oversight on this kind of research. But can you give me 2 or 3 things that this virus has that make you think it's lab versus some of the evidence for MERS and SARS that it came from animals?
Yeah, there are 3 regions: the receptor binding domain, the furin cleavage site, and this protein 8th from the left called ORF8. With respect to the with receptor binding domain, if you look at what happened with SARS-1, we have the virus sequence when it first was in civet cats in the markets, jumped into a few humans. We have the virus sequence then. It started infecting more. And then we have the virus sequence when it became, when a human-to-human passage could occur and an epidemic occurred. And so you can see the progression of mutations as the virus adapted from being in civet cats and then being in humans. The first jump into humans, it had only 15% of the mutations it needed to support an epidemic. Okay, let's flip to SARS-CoV-2. When you look at the virus that first entered the human population, out of all of the changes in the receptor binding domain, there are 200 amino acids, 4,000 possible changes. There were only 17 mutations that could make it a better virus. Its receptor binding optimization was 99.5%. And in fact, one of the 17 ended up being the Delta variant. So that kind of optimization juxtaposed by the fact that there were no patients in Wuhan, 36,000 blood bank specimens tested for antibodies in 2019. Not a single patient was infected. Let's go back to SARS-1. 20% of all people in the markets were infected while the virus was practicing to set up an epidemic, 1% of the general population. So we would have expected 360 in the general population. In Wuhan, we had 0. Furin cleavage site has obviously never occurred in this related viruses, sarbecoviruses, that split from their cousins, the MERS viruses, around the time of William crossing the channel, 10,060. That was when the sarbecoviruses came. So there's never been a furin cleavage site, and the genetic sequence of it uses a code that's never been used, the CGG dimer as it's called, which has never been used before. Finally, ORF8, this protein that goes into the bloodstream and suppresses interferon response, so you're asymptomatic, and suppresses AHC antigen presentation, so you can't make good antibodies. This was the subject of 2 master's theses at the Wuhan Institute of Virology. I have found no Western scientists that worked on this location in the genome before 2019. And the protein is not present in MERS. It has a 5% homology in SARS-1. Between SARS-1 and SARS-2, there's a protein there, but it's only 5% homologous. But this master's thesis, the first one, optimized its function in suppressing interferon, symptoms of fever and chills, and suppressed its antigen presentation. The second one was making synthetic biology tools so you could move it around inside genomes.
So to reiterate, there's been no animals found that have COVID-19. When they did find that animals had the first SARS and MERS, they found it out within months. When they tested the animals in question, 90% of the animals had the SARS virus. So we haven't found any animals yet with COVID-19. And then most viruses that come from animals first aren't very infectious at first, and they infect a few humans. So you don't have a pandemic that does this. It smolders and then does this. During the smoldering phase, you find background antibodies that people have had it, even if they don't know they had it. So when they tested the background of people who were working with the animals that had COVID, they found 20% of them had antibodies to having had SARS.
SARS-1, yes, correct.
But then if we test the people in the marketplace, we're not finding that. If we look at the people in the Wuhan marketplace, we're not finding significant numbers that were positive and finding almost nobody positive from the previous year that had been ill.
No, it's 0 out of 36,000.
Thank you. Why don't we do a second round, and why don't we start, we'll go the same order. Senator Johnson?
Dr. Quay, how did we find out about the Nipah virus?
In December 2019, 5 patients at a Wuhan hospital had their specimens sent, a bronchial lavage, where they stick a throat and get it lost, to the Wuhan Institute of Virology for sequencing. The process is to amplify it with a PCR process. You make a lot of copies of what's in the specimen, and you usually inadvertently make copies of what's going on in the laboratory. So the Wuhan Institute of Virology probably regrets, but they put a 55 million letter database of the background information up in the gene bank, which is the NIH's database there, of everything going on. We found 20 strange things in these patient specimens: honeysuckle genes, horse viruses. 19 of the things we found were in publications from the laboratory over the previous 2 years. So this clearly was a signal of what was going on in the lab around there. The one thing they didn't publish on was this cloning vectors of the Nipah virus. So it's in the patient specimens because it was in the laboratory at the time, not in the patients, but and they have never published on that at this point in time.
How do we know it's 60% lethal?
Oh, the Nipah virus has had sporadic epidemics in the belt around Africa and India, Bangladesh, and it's between 60 and 80% lethal in the pockets where it comes out. It's not very transmissible like Ebola, so it kills 100 or 200 people and then burns out. But if they made it airborne, it would be different.
Okay, so this is a virus that occurs in nature, but you just detected it in this database, okay.
I detected cloning vectors with it, so they're manipulating it, which is not allowed by biological treaties.
So, and that's a pretty scary scenario right there, that the Wuhan lab that might have been the originator of the coronavirus is fooling around with something far more deadly.
Yes.
And they're, obviously, mum's the word.
So we'll go to Dr. Quay next. You may be familiar with the genomic sequences in NIH's database. I think you spoke about them, that Chinese scientists asked to be removed and how they were from early COVID Wuhan patients. Do you believe there could have been more data in NIH's database submitted by Chinese scientists that could hold the key to the COVID-19 origins?
Yeah, this was a really nice piece of work by Jesse Bloom at the University of Washington, who found not in the NIH database, but on some Amazon web servers, the actual sequences of viruses from very early patients that had been put on gene bank and then removed before they were published and made available. And the remarkable thing is, again, going to another piece of good research, the virus that first came out, the first Wuhan virus, is 3 mutations away from what we now know is probably the first virus, but that's a computational method. It's kind of complicated, but anyway, there's a prediction. There are 3 mutations that have never been seen in humans before the first virus that we have in humans. The specimens Jesse found had some of those. So we know that the Chinese have viral sequences that are ancestral to what we have, and the more of those we get, the more we'll get to the bottom of this. I'll point out that these sequences were from September and October of 2019, 2 months before any person in the market was sick. So again, the timing of the market spillover doesn't coincide with the genetics of the virus.
For 20 years, NIH sponsored EcoHealth's partnership with scientists from the Wuhan Institute of Virology. The Chinese scientists have bragged that their virus sample database is the largest in the world. They took that database offline in September 2019. NIH asked EcoHealth for research records. EcoHealth told them that the records are in the custody of the Chinese government. Is it possible that the database taken offline by the Chinese government was data collected by EcoHealth and belongs to American taxpayers?
Make sure they have my speech.
And Dr. Quay?
Well, since the work has been funded in part by U.S. taxpayers, then by definition, access to that would be important. And I also think that we don't have to rely on the Wuhan Institute of Virology for releasing that. I believe within the U.S. jurisdiction, there will be copies of that database. It's too valuable not to have in your own possession if you're doing research on it.
Do you think there's any way we can still get any of that data that's missing? I feel like, you know, somewhere we're going to find the grandfather of COVID or a cousin or something here in these data banks. Why did they take them down? And I mean, what would be the advantage of them taking it down? Do you think we can ever find what we're missing?
Well, it was taken down at 2 a.m. on September 12th, 2019, which is, I mean, I guess everyone works hard, but that's a little suspicious to be doing it at that point in time. I believe it contains closer precursors, and my hypothesis is it contains the one that's 50 mutations or 100 mutations, not 1,200 away, and it was too obviously a smoking gun. But again, if you're collaborating on that and you're spending 10 years building a database inside the Wuhan Institute of Virology, you're going to mirror that database in your own facilities, which means that it's got to be at EcoHealth Alliance somewhere.
Thank you all for appearing.